go ahead.
All right, sorry guys, I forgot to record till now, but this is the first slide, so we're good.
Yeah, this is just remember that microtubules are embedded in the MTOC at the minus end, and they grow from the plus end. And then we have our centromeres versus centrioles. This is the centrosome matrix, as you can see there, and the pair of centrioles are in the middle. And the gamma tubulin ring complexes are these little red rings. So yeah. Okay. So the biggest thing with microtubules that Laura likes to talk about is dynamic instability in the GTP binding pocket. So both the alpha and the beta subunit contain a GTP binding domain. The GTP binding pocket on the alpha subunit faces the positive end of the microtubule. When the beta subunit binds the alpha subunit containing a GTP molecule, it covers the GTP binding pocket of the alpha subunit. And the beta subunit GTP binding pocket also faces the positive end, so both of them face the positive end. And the beta subunit has GTPase ability, so it can hydrolyze GTP to GDP. And these are the subunits of the tubulin. And the ability of the tubulin heterodimer is to hydrolyze GTP allows for the dynamic instability of microtubules. So we'll get more into like the diagrams of how that happens, how they grow and fall apart. So dynamic instability describes the method in which microtubules use to grow and shrink to adapt to the needs of the cell. You need them both to grow and shrink. If you can't grow, then you're going to have a problem. We're going to talk about drugs that inhibit these processes and why they would need to inhibit them, but that's later on. So dynamic instability mainly is governed by GTP hydrolysis in the beta subunit at the plus end. So that's the biggest takeaway. GTP hydrolysis beta subunit at the plus end. Okay, so it's like the biggest part of dynamic instability, but we'll talk more about it in the next slide. So this is just the diagram that Laura showed in class, microtubule growth and shrinkage. The ability for beta tubulin to hydrolyze GTP constitutes microtubule growth. So when you have beta tubulin plus GTP, you're going to get growth and stability. You're going to have more stable ends, and you're going to add to the end. So this first, the A section of the diagram where it says growing microtubule, you have beta tubulin bound with GTP. So the GTP tubulin molecules add to the end of the microtubule. Remember, this is at the plus end, because at the minus end, this is where they're embedded in the microtubule organization center. And then the addition proceeds faster than GTP hydrolysis. So by the time that GTP hydrolyzes into GDP, you're adding more already. And then you get this GTP cap, which is basically this GTP bound to beta tubulin. Yeah, so this red is the GTP cap bound to beta tubulin, and this keeps the microtubule stable. And then we have on the other end of it, the shrinking microtubule is what happens when beta tubulin hydrolyzes GTP, because the beta tubulin has GTPase activity. So basically, they peel away from the microtubule wall when tubulin is bound to GDP. And then GDP tubulin, beta tubulin is released into the cytosol. This still is happening at the plus end, and basically think of it as going from the plus end, shrinking down to the minus end. And then on this side, you have the minus end growing with the plus end is growing from the minus end. So we put the little labels here, but where it's peeling away is the plus end, and where it's adding is also the plus end. And if tubulin hydrolyzes GTP faster than it's added, then there's no cap, which also leads to instability and shrinkage of the microtubule.
Awesome. So the next thing we're going to talk about is how we study microtubules. So you could look at microtubules in vivo, which means like in a living cell or in a person or something, or you could look at it in vitro. First, let's look at it in vivo.
Wait, can I just
So,
say one thing real quick? Sorry.
yep, yep.
A good way to remember in vivo versus in vitro is I like to think of as in vitro has a T in the same as like petri dish, so it's like not in the living cell. Yeah, so in vitro is like think of it as like in the petri dish, not in the living cell. Sorry, that's
Yep,
my one
and
spiel.
vivo is
Could you
kind of
repeat
like
again
this.
what vivo is?
Yeah, it's like in life pretty much. So vivo is
Yeah,
like from like the Latin word life, vivir, or Spanish, I guess. So in vivo means like in a living thing. So like in a person, in a cell, something like that, as opposed to in a petri dish, like Rachel was saying, for in vitro.
in vitro. Yeah. So if you see in vitro, it's being done outside of a living organism, and it's in like a. Just think of the T as petri dish.
Okay, thank
Hmm.
you.
Yep.
So, okay, so let's first look at it in vivo. So in a living cell. So in vivo, there is only growth and shrinkage at the plus end. Can any of you guys think about why that might be?
Because
Either,
if it's coming,
yeah,
sorry,
go for it. Go for it.
because if it's coming from the minus end, then the growing and shrinking is too slow. Whereas in the plus end, then it's goes a lot faster. And in this case, the minus end is plugged into the gamma tubulin ring.
Yeah, exactly. More the second end for in vivo. So the second part of what you said, I mean, so in vivo, the microtubule is inside this gamma tubulin ring, so nothing can get at the minus end. So the only place where you can add is at the plus end. Does that make sense? So you can't, like, nothing's going to happen at the minus end because it's like protected inside the gamma tubulin ring, and nothing can get to that. This is as opposed to in vitro. So in vitro, all the microtubules are just floating around in a petri dish. So that means that they're not using a microtubule organizing center. They're not plugged into a gamma tubulin ring complex. So that means that growth and shrinkage can happen at both the plus and the minus end. So in this case, addition and depolymerization can happen at both the minus end and the plus end. But in this diagram, the size of the arrows represents like the rate of the addition or depolymerization. So the bigger arrow for addition means that the addition is happening way faster at the plus end, and so is the depolymerization to an extent. It's interestingly, both happen at faster rates on the plus end. So even though the minus end is where things shrink to, like depolymerization is still a little faster on the plus end. Okay, so a lot of the questions Laura asked will be in an in vitro assay because that's the easiest place to study a microtubule. So what are key factors when you're putting something in vitro?
Whatever's happening on the outside of the cell has to be physiologically possible within the cell.
Yep, that's definitely true if you want it to be like physiologically relevant. But I'm think I'm thinking so what, like for an experiment, what are the key factors of the experiment um that are only true in vitro and not in vivo?
Would it be
Is it not connected to the gamma ring?
Um yeah, that's one of them. Does anybody else have another
what about
suggestion?
the concentrations of like
Ye-
what's there? Because like we can adjust concentrations outside the cell.
Yep, exactly. So in vitro, there is a limited number of tubulin, so the things that get added to the microtubule, and there's also a limited number of GTP. And if you remember from what Rachel was talking about, that you need GTP tubulin needs to be bound to GTP in order to add to the microtubule. So if and eventually when you're depolymerizing, GTP gets hydrolyzed to GDP. So if there's nothing, there's only a limited amount of GTP, then eventually all of the GTP is going to get used up. So eventually you won't have any addition at all, and the microtubules will just completely shrink it entirely, and they'll just disappear. That's like what will happen eventually in all in vitro assays if you don't add GTP. So it'll be normal at first, but eventually it'll just like the amount of tubulin will just decrease sharply. Any questions on that? If not, we'll go to some more details on the in vitro assay. Okay, so in vitro assay. So what we're doing in an in vitro assay is we're measuring the amount of polymerization, like the amount of addition to microtubule, not like the total length of the microtubule. So how we do this is with a spectrophotometer. You might have used these in like a Bio 153 lab or maybe Orgo lab if you've taken that yet. So what a spectrophotometer does is it measures absorption and refraction. So if you have more microtubules in the test tube or petri dish, whatever you're using, when you use the spectrophotometer on the test tube, then more light will be absorbed. So like if there's a lot of microtubules in the test tube, then like the microtubules will absorb the light and you'll get a higher absorbance reading. If there's more just tubulin, then those are kind of like invisible, so they're like soluble, I guess. Is probably the reason. So more light will be refracted and you'll have a lower reading. So if you have more polymerized microtubules, then more light will be absorbed and you'll have a higher reading. If you have just tubulin dimers, so like the things that are getting added to the microtubule, you'll have more light refracted and a lower absorbance reading. Okay, so if you plot if you plot the absorbance reading from the spectrophotometer on a graph versus time, you'll see this. Sorry, excuse me. You'll see this curve. So in the beginning, you have a lag phase, and that's when the individual dimers, the tubulin dimers, are trying to organize together and form. This takes some time because they have to like float around in the test tube and bump into each other in order to form. So this takes some time, and that's why it's called a lag phase, because this takes like one of the longest times before it actually starts doing anything. The next phase is the elongation phase, which is this part right here. And that's when you're having the addition of tubulin at both the plus and the minus ends. Remember, this is an in vitro assay, so you add it to both the plus and the minus end. Eventually, the rate of tubulin addition and depolymerization will reach an equilibrium, so you'll have this plateau. So there won't be any, like, it'll add and subtract at the same time while there's still GTP available. And eventually, like so the absorbance reading will stay about the same, and it'll be this flat line. So what isn't shown on in this graph is what happens after. So what do you think what would happen in this graph if you had a limited amount of GTP? What would happen to the absorbance reading?
If you have a limited amount of GTP, what would happen?
I would assume that the elongation phase wouldn't have as steep of a slope. So, and it's possible that the lag phase could also be a tad bit longer.
So say you have I worded this poorly. Say you have enough GTP to get to the plateau phase, but then eventually you run out. What happens to the graph? Like everything's normal until the plateau phase.
Wouldn't it just start, like everything would begin to unravel and you have catastrophe. So then I feel like your graph should start to decline.
Yep, exactly. So like you said, so there'll be a what's called a catastrophe event, which is when microtubule starts depolymerizing. So eventually the graph will start at like a steep incline down this way. So it'll kind of look like almost like a bell curve, like it'll even out and then sharply decline down.
Because
back
remember
to,
that this occurs in a closed system. So there's a finite amount of tubulin and a finite amount of GTP. So over time, if you let the experiment go long enough, GTP is going to be hydrolyzed to GDP, because remember, beta tubulin has that ability. So then if you let it go long enough, all the microtubules will depolymerize due to all the GTP being hydrolyzed.
cool. So any questions on this? We'll have some practice questions at the end on this, but if not, we can move on to different drugs that affect the system. Okay.
So I love this because I actually take colchicine, fun fact, and I never really knew how it works until I took this class, and I was like, oh my god, other people know about it. Anyway, so these are the 3 drugs that Laura talks about. You have Taxol. Taxol is a anti-cancer drug. It stabilizes microtubules, and which means there's no depolymerization. This prevents the microtubules from peeling away and going through that catastrophe that we were talking about, how it will shorten basically from the plus end. So this stabilizes it. Colchicine binds subunits and prevents polymerization. So both colchicine and vinblastine prevent microtubule polymerization, which leads ultimately to microtubule disassembly. And colchicine's used to control inflammatory responses. So for me, and I'm kind of putting myself on blast, but I have a autoimmune disorder. So I take colchicine to combat the inappropriate inflammation that occurs, and it basically prevents the microtubules from polymerizing, which leads to more instability. And ultimately, I did a research paper on the effects of how that ties into inflammation, but you don't really need to know that. It's very specific to just colchicine. But vinblastine is also used for cancer. It's for use in chemotherapy, and it also prevents polymerization. So Taxol prevents depolymerization, and then you have colchicine and vinblastine that prevent polymerization. Does anyone have any questions on these drugs? So you have 1 stabilizing drug and 2 destabilizing drugs. Okay. Cool. So motor proteins, we talked about this in class today a little more. Motor proteins are fun because they look funky and they look like they're walking. And I don't know if you guys seen, but there's a lot of funny videos out there of just motor proteins. So the 2 main ones you want to know are kinesin and dynein. They're both ATPases, which constitute their movement along the microtubule. Their main job is to transport cargo around the cell on the microtubule tracks. And Laura talked a little bit today about how microtubules are usually like 13 or something long. It's not just one like line of tubulin. So dynein is a multifunctional minus end directed motor, which this is at the bottom, kind of skipped ahead, but dynein walks towards the end of the microtubules, the minus end, and kinesins walk to the plus end of microtubules, except kinesin 5, which is the only kinesin family that you need to really know, besides 13, which is they go to the plus end ultimately. Kinesin 13 goes to the plus end, but kinesin 5 is the only kinesin family that will go to the minus end. Other than that, just remember that kinesins go to the plus end of microtubules, and dynein goes to the minus end of the microtubules. They also play a big role in flagella and cilia movement, which is more not so much our cells, except for like reproductive cells, but yeah, they play a big role in that. So they're usually plus end, kinesin, and dynein is negative end. She goes a little into more detail about the families. I don't know if, because when I took her class, we didn't have to know the specific families. We knew like kinesin 1, kinesin 3, kinesin 13. That's not on the next slide, is it, Jason? Oh, okay, it is. Yeah, so these are the different kinesins and their families. So we have 5, 1, 3, and 13. So remember that 5 is the one that goes to the minus end. So also, a thing to remember, kinesin 5 is most kinesin family members are dimers, but kinesin 5 family kinesins are dumbbell shaped and they're tetramers. So it's the only kinesin family, kinesin 5, that is a tetramer shape instead of a dimer. And you can see in this graph, you have the tetramers on either side, and then you have these dimers for all the others. So that's the biggest difference in kinesin 5. And basically, kinesin 5, how it works is 4 subunits bind in the way that the motor heads are at each end of the kinesin, hence the dumbbell shape, as you can see here, and in the inactive and active, but we'll get to that. And so having the motor domains at either end allows kinesin 5 family members to bind to microtubules at either end and slide the 2 microtubules like along, like it slides the 2 microtubules like, I'm trying to, how to explain this? So see the arrows, the directional. So this one for kinesin 5, it's sliding to the right, and then the top is sliding to the left. That's what it means by it's binding to 2 different microtubules, and this kinesin 5 is special. It has that ability because it has that tetramer shape and this dumbbell conformation. And so this allows it to bind to 2 separate microtubules and slide them, which is kind of important in spindle assembly, also in cell division, but it's kind of just a perk of kinesin 5 that it can exist. Then we have kinesin 1. So we're back to dimers. It's a plus end directed motor, and it moves cargo from the middle of the cell to the periphery, which means it moves it from the middle to the plus end. Basically you have this cargo and it's going towards the plus end. Then we have kinesin 3, another dimer, also plus end directed motor. And this is mostly involved in movement of cargo in neurons. That's why you have your synaptic vessels. But basically they're simple. Kinesin 1 and 3, they transport cargo to the plus end. Kinesin 3 is in your brain. And then kinesin 13, also dimer. They don't use their motor domains to walk along the microtubule like kinesin 1 and 3. Instead, they diffuse laterally along the microtubules until they reach the end of it. This is where their ATPase domain is activated, and they use their ATPase activity to depolymerize microtubules. So you can see here is destabilizing the plus end of microtubules. So this tubulin is peeling away. Any questions on these kinesin?
Okay, if not,
Oh wait, one more thing I just wanted to add, sorry.
Mm-hmm.
The main thing also with motor proteins is their movement is that all motors, including dynein and kinesin, are ATPases. They use the coordinate binding of ATP, ATP hydrolysis, ADP bound, and ADP release to basically direct their function and how they move. So one of the movies, I think it's 17.7, you can see how kinesin 1 movement is regulated, which is the second one, towards the plus end with cargo. And in the ADP bound state, the kinesin motor domains do not have a good affinity for the microtubule. So basically ADP inactive can't bind really well to the microtubule. The kinesins diffuse around, and when a motor head makes contact with the microtubule, the motor head changes conformation, which allows for ADP dissociation and then rapid association of ATP in the motor ATPase domain. So once it has that ATP and its ATPase domain, the motor head has good affinity for the microtubule, and ATP binding causes the like neck linker region, which you can kind of see in some of these. Yeah, Jason's kind of pointing to it. To swing the other motor domain forward. So when you see in the video, it literally looks like it's walking, and it's because those two domains are swinging each other forward. And with constant ADP being swapped out for ATP, and then ATP being hydrolyzed, and then ADP again, it's just this cycle. It's a lot easier to see in the video, but that's basically how these work.
Yeah, definitely check out that video. It explains it
Yeah.
super well.
Yeah, definitely.
All right, if there's no questions on this, we can go to microtubule associated proteins, or MAPs. The only one she talked about this year is tau, which is highly expressed in central nervous system neurons, especially axons. So in the central nervous system, you want microtubules to be super stable because a lot of so there's lots of microtubules in the axon that transport proteins down to the synapses over here. So you want the cells to be super stable so you can do that. And what tau does is it binds microtubules and stabilizes them. Tau itself is incredibly heat stable. It's hard to denature, but conveniently it's easy to purify because it's hard to denature. Excuse me. So what happens when tau goes wrong? So when tau is messed up, which is the case in many neurodegenerative diseases like Alzheimer's and sometimes Parkinson's and stuff like that. If tau becomes hyperphosphorylated, so if it is phosphorylated too much, it won't bind microtubules. So when it doesn't bind microtubules, it like tangles itself up, and these tangles can spread throughout the brain and cause a lot of problems, which is what eventually leads to Alzheimer's. So a lot of research is happening right now, figuring out how that happens and how to fix it. Any questions about that? She
I have
probably,
a quick question.
yeah. Mhm.
So when we're talking about these proteins and everything that are essentially stabilizing microtubules, does the stabilizing of microtubules just mean that like these microtubules are undergo shrinkage, that it'll stabilize that polymerization?
Yeah, pretty much. So when you talk about stabilization, you usually mean that it's not depolymerizing. So it's like staying the same length, or it can grow if it needs to. I think. Is that right, Rachel?
Yeah.
Okay. Yeah.
Yeah.
So anytime you talk about stabilization, it's mostly just avoiding depolymerization. And
Yeah.
Okay, I just wanted to like make sure, just because I didn't know if that in and of itself could cause problems within the cell, like because since it won't ever be able to shrink, because I know the cell has shrinkage for a reason.
Mhm.
Right. Yeah. There's definitely cases where you want to shrink. We'll talk about that next week a lot in
Yeah.
cell division.
Okay,
So you'll see that next week.
okay, thank you.
Yep. Okay. The last thing is this really cool protein called katanin. It's named after a ninja star because it looks like a ninja star, which is super cool. I love it. It's not. I don't think she'll ask you like what it is, but she might like give you a problem where like katanin is used. What happens? Something like that. So what it does is it just like chops up microtubules. So you could use it to just mess up a microtubule, chop it up, and then we'll see it next week, I think, when we're talking about spindle fibers mostly. And you'll see it's regulated at the beginning of the end phase of the cell cycle by mCDK. So this is a kind of like a preview for next week. She mentioned it briefly this week, but we'll talk about it more next week. Okay, so let's do some
questions.
practice questions. Woohoo! We can go back to Pirates of the Caribbean. Okay, so first question. Kinesins and dyneins both have what? Or are what? You can answer in the chat or unmute, either way.
Yeah, there's only 6 of us, so don't feel. Got B, 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 B. Yep, B.
Yep, that's exactly true.
They derive
So
their energy from ATP hydrolysis. Colchicine and Taxol are more focused on microtubule stabilization and destabilization. And yeah, so those are incorrect. So it's not D and it's not E because B is right.
cool. All right, number 2, hydrolysis of GTP to GDP is carried out by tubulin molecules. And this does what?
Anybody? What does hydrolyzing GTP to GDP by tubulin do to the microtubules? We got an A, we got an E. So 2 is actually E, mainly because this is the main thing. The whole GTPase activity of tubulin is what leads to this dynamic instability. It wouldn't be A because it doesn't provide the energy because tubulin already has that intrinsic GTPase activity. That's the reason I would not say A. I don't know, Jason, if
I
what you think.
don't exactly know. I just know that's not the case.
Yeah, I would say it's because of that GTPase activity. Because it has that intrinsic ability anyways. That is a little tricky though, because I mean yeah. But it is E. And then D, C, and B are not correct because hydrolyzing GTP to GDP is not assembly, it's disassembly. Yeah. So that's kind of why 2 is E. Does anyone have any questions on that one?
I just maybe to help explain maybe why not A is since it is disassembly, you know that it's not going to promote polymerization, but more so
Yeah.
active depolymerization.
Yeah. That's a good explanation because they're hydrolyzing GTP to GDP. So you're going from that active to inactive state, which is falling away from the microtubule. So that's that depolymerization. Yeah, I didn't see that polymerize at the end of it, but so yeah. Good. Okay. 3, a growing microtubule has what? What do we think? I wish we had like Jeopardy music.
So lame.
I know.
Anyway.
Do you guys was there supposed to be a specification as to what end? Or
No, just at the end.
okay.
So we have an A, yep. A, good. At the end, what end does this occur at? Follow-up question.
The plus end?
Yep, so it's not going to be throughout the microtubule, it's only at the plus end, and it's not GDP, so it's GTP when you're growing.
In vitro could technically occur at the minus end too, but mostly it's at the plus
Yeah,
end. It's much faster there.
That's true.
That's why I didn't put N, because I didn't say in vitro.
It doesn't
either.
specify in vitro or in vivo, yeah.
Yeah.
But in vivo will be at the plus.
Mm-hmm. All right, number 4, for a given population of microtubules undergoing dynamic instability, which is true. So you have a bunch of microtubules in a test tube. What's happening?
Typo
don't
to
think-
shore
I don't think
in.
it's H.
It's H.
On the slide, like for whatever reason, like maybe I'm just
Get some D's.
Yep, all
Yep.
D's. That's correct. So at any given time, some will be growing, some will be shortening. Like remember, it's like a dynamic process, so not all of them will be happening at the same time. Any
Yeah, I think one of the
questions?
videos shows it. It's like pretty cool, like the animation where the like some are going out, some are going in. Yeah,
Mhm.
probably one
Yeah,
of the movies.
definitely watch the animations in the movie folder for these this week, especially, and the coming weeks, because they're super
Yeah.
helpful.
Yeah, definitely.
Okay. So most of these questions use this graph. But first, we talked about this a little bit. What is the difference between in vitro and in vivo? Not like the definition, like what's specific about each type. Remember, we said in vitro was in a petri dish, and in vivo is like in a cell.
Do.
You don't have any guesses?
like mentioned before, like in vitro, which is outside a living cell, it can add on both ends. It's just the rate of addition will still be greater on the plus end. Whereas like in vivo, since the minus end is embedded into the centrosome, like with the gamma tubulin, you know that like no addition can occur. So in the cell, it can only have GTP tubulin bound addition on the plus end.
Yep, exactly. And remember, we said in vitro also there's a limited amount of tubulin and GTP, so that could have effects. All right. So number 6, what would happen if you added? So this is an in vitro reaction. What would happen if you added gamma tubulin to the reaction? This could be multiple options. So remember, gamma tubulin is part of the microtubule organizing center that helps like get a microtubule started.
Here's one B. B and D. A and D.
Good, we have some.
And maybe B and C.
All right, we have some contention here. So, so I would say that the answer would be a shorter lag phase, so a longer elongation phase, and definitely E, because the max absorbance would be larger.
Yeah.
So the reason I would say lag phase is shorter, I saw a bunch of people said B. The lag phase would be shorter because if you're adding these gamma tubulin ring complexes, so or gamma tubulin, that would allow the microtubule to form faster. So so normally the lag phase is like the amount of time like the pause it takes like for microtubules to get started. If you add a gamma tubulin, that process would be faster because gamma tubulin helps to organize microtubules. So if you added that, it wouldn't take as long to organize themselves, and then the lag phase would be shorter. Does anyone want to explain why they thought the elongation phase was longer? I actually don't have a great explanation for that. Rachel, do you? I think it's just because that there'll be more like I don't know.
Yeah.
They'll form faster, and then so that means that they'll be able to be polymerizing for longer, I guess. So if they can form these complexes faster, then they have more time to polymerize before they start depolymerizing.
Yeah, that's what I was thinking.
And then E, the max absorbance would be larger. So we said that absorbance is larger when there's more microtubules like in complex. So when you add this gamma tubulin, you'll have more microtubules in complex because they can form faster and easier. So you'll have a larger max absorbance. Any questions about that?
Still a little confused on the elongation phase as to like why it would be longer. Like, wouldn't adding like a base essentially allow elongation to occur faster, essentially like speeding up the elongation phase, thus making it shorter?
Yeah, I honestly don't have a good explanation for this. I took this from somewhere else, so I don't I don't have a great explanation. Like my previous explanation is the best I got. I might check with Laura on this one though, because
Yeah.
I could definitely see that too. So I'm not completely sure. So
Yeah, and
but,
a lot, these are like the type of questions like she's gonna probably ask if you see these on the exam. But yeah.
So yeah, the ones that we're definitely sure about are the lag phase would definitely be shorter and the max absorbance would definitely be larger.
Yeah.
I'm gonna check on this one because I don't have a great explanation for it. Okay.
Yeah.
So same choices for number 7. What would happen if you added Taxol? So if you remember, Taxol is a microtubule stabilizer.
ACF because Taxol is a stabilizer.
Any other guesses for this one?
Definitely F. All right. Well, this one, the answer is actually C and E only. This stuff was never my strong suit, if you want to explain, Jason.
I would say there's definitely no lag phase because Taxol doesn't affect anything for like organizing it. Taxol would affect it once it's already formed. So I don't think lag phase would be affected here. Yeah, the stuff's tough, so don't worry about it. So I would say that max absorbance would be larger. I don't know exactly what max absorbance would never be reached means. I think that's like a fake answer just to distract you.
Yeah, I don't. Yeah.
So I just think it's
like mistaken. Max absorbance never reached for like plateau would never be reached.
Oh yeah, I can see how you think that.
Okay. Because
yeah,
if Taxol is like constantly stabilizing, you wouldn't have equilibrium be reached.
Right. Exactly.
And it would never, like, elongation phase would essentially just like continuously occur. So like, would it elongation phase? Would that make elongation phase longer? And are we talking about elongation phase in terms of time or in terms of like the velocity?
In terms of time, so it'll mean that this graph has like a less steep slope, so it'll take a longer time to reach plateau if it ever does. So yeah, I would definitely agree that the elongation phase is longer because if Taxol is stabilizing the microtubule, then you can just keep adding until you run out of GTP or if you run out of tubulin dimers to add. So it'll just keep getting longer and longer.
And then
would be-
Taxol, what does it do for the lag phase? Does it do?
I don't think it does.
It doesn't
anything during-
affect it.
Yeah, it doesn't change it because it still has to form, and Taxol doesn't really
Right.
do anything until microtubules have already formed, right?
Yep, exactly. Taxol will affect an already formed microtubule, but it doesn't affect the formation of the microtubule. Okay. So number 8, if you let this reaction, so no drugs or anything added to it, just a standard reaction with tubulin and GTP. If you let this reaction run for a long period of time, what would the graph eventually look like?
The graph would decline because it will eventually run out of GTP.
Yep, exactly. So it'll look normal, it'll plateau for a while, and then once it runs out of GTP, it'll go woo and go back down to 0 absorbance eventually. Cool. Why is because you said because the microtubules will run out of GTP and it'll eventually polymerize. Alright, so I'll take the answer key.
Only one more slide of 2 questions.
And then we can go back to Pirates of the Caribbean. All right.
So we
Number
know
9.
Taxol and vinblastine interfere with microtubule assembly. Why do they make effective chemotherapies? So what do you guys think?
This one's tough because we haven't talked about cell cycle yet.
Yeah,
So I'm just gonna go over real quick.
go ahead. Just say.
So, mitosis requires spindle assembly and disassembly. So if Taxol is stabilizing microtubules, so you have more assembly. Vinblastine is destabilizing the microtubules, so have more disassembly. And if this isn't like perfectly regulated like it is in normal cell division, then the cell will have problems and it won't divide. So that's in cancer, you have problems with cell cycle. So if the cell cycle is messed up, then it won't divide and you won't have these cancerous cells.
Yep,
And so that's
Sophia,
why
you got it. Prohibit cell division is their main through the cell cycle, which we're going to go over the cell cycle next week. And we talk a lot about cancer and P53 and all that stuff during the cell cycle lectures. But yeah, so because the main problem with cancer is you have cells that are dividing too fast that are not, that are mutated or unhealthy cells. And their main role is to kind of stop that.
Yep, exactly. All right, so last question. In cells, dynamic instability typically occurs where? Remember,
so what?
in cells, so in vivo.
Mm-hmm. So yeah, so it's A because when you see that in cells is equivalent to in vivo. Yeah, you guys got it. Remember that unless it says in vitro with that T petri dish, then that's when you get at both the plus and the minus ends, but this is in vivo in the cells and that occurs only at the plus ends.
All right, that's all we have. Does anybody have any questions about anything we went over? I will check on that question with the elongation phase because I don't have a good answer for it, but anything else? If not, thank you guys for coming out. Have a great night. Feel free to email us with any questions as always.
Yeah. Have a great weekend, everybody.
Thank
Good
you.
weekend.
Thank you guys for hosting these. Really
Thank
helpful.
you.
Yes,
No
thank you.
problem.